Our next endogenous hormone is one that is an outright growth inhibitor. And although auxin can be either a promoter or an inhibitor, abscisic acid is usually thought of as being, generally speaking, growth inhibiting. So we'll label it as an inhibitor as far as growth is concerned. Abscisic acid stimulates the process called abscission. And we talked about it before, but let me make sure everybody remembers by mentioning what it means. Abscission is basically, I've used the term self-amputation that plants will use to drop leaves or maybe fruit or flower parts that are no longer useful to them. And basically, it doesn't, maybe a less gruesome way to word it would be dropping parts, leaves, flowers, etc. That's what abscission is. And ABA, for short, or abscisic acid, stimulates this process. They are synthesized in any cells that have chloroplasts, so mainly young shoots and leaf cells. And as it says in your notes, they're often produced in response to some environmental stress. And as a result of the stress, the ABA triggers certain plant adaptations in an attempt to deal with the stress. And some common plant stresses that cause an uptick in the production of abscisic acid in plants are things like flooding, excessive drought, nutrient deficiencies. In other words, the plants are starving to death. Things like salinity, salt water, irrigation water there's an I in there. Salinity problems, too much salt in the irrigation water, or perhaps from storm surges. And just generally speaking, other types of injuries, damage due to windstorms, or maybe to animals eating them, or being damaged in one way, shape, or form physically. In response, the plants produce ABA, which causes the following effects. It is often involved in maintaining bud and seed dormancy, which, although not necessarily a result of a stress, this is a means of seed survival in that dormancy helps a seed avoid germinating in the wrong time of the year. For example, a season that is too dry or a season that is too cold for the seedlings to survive. It's obviously involved or triggers the abscission, as we said above, or dropping of plant parts. And it has also been shown to be useful in plants during drought or wilting type conditions to cause the stomata to close. So it's used to regulate, we can say, guard cells. And we'll say that stimulates stomata closing, or triggers it. Next hormone on the list is another inhibitor, and it's got something else that's fairly unique about it, and it's called ethylene. Ethylene has a chemical formula of C2H4, and basically it's small enough to draw. It looks like this. There's a double bond between two carbons, and each carbon has an additional two hydrogens connected to it. What's unique about it is that ethylene is a gas. These others are liquid or some sort of acid or salt sort of compound that is dissolved in the cytoplasm, for example, or in xylem fluid or phloem sap, where ethylene is a gas. And ethylene gas, as it says in your notes, is not particularly water soluble. And so the parts of the plant that ethylene will stimulate a response in usually have to be very close to the part of the plant that actually produces the ethylene, because it doesn't get transported in xylem or in phloem. And for the most part, the ethylene, we'll say, works in the cell or nearby cells in which it was produced. In other words, there's not long distance transport of ethylene gas within the plant. So what are some effects? Well, I mentioned ethylene is considered a growth inhibitor, and it has the following effects on plants. Number one, it dramatically increases or causes an increase in respiration. And while we think of things being that respire as being alive, and respiration, if you're a living organism, is definitely a pretty good thing in most cases, if a plant dramatically increases its respiration rate, it can respire or burn more sugars than it is actually producing, and the plant can start to go into decline, like we talked about before with creeping bentgrass decline, for example. The problem with that was, once again, the plant respires more than it is photosynthesizing because of the effects of photorespiration. So this would be therefore considered a negative effect in terms of growth. It is also a cause of leaf abscission. It's actually used to defoliate certain crops to make it easier to harvest the fruit from them. And cotton is a good example of that. They'll use ethylene producing sprays to defoliate to make it easier to get at the cotton bolls. We've also got effect of, or possible effect with ethylene, of flower abscission. And it's also part of, or triggers, fruit ripening. And this is taken advantage of in some cases. For example, some crops are picked much earlier than the time in which they would be ripe, and so they're picked, let's say, green. Through the harvesting process, some of the cells on the outer portions of those fruits can get bruised. And let's say we got a green tomato and it's picked green. Well, you pick enough tomatoes and you put them all in crates. Some of them are going to get bruised even just slightly. And those cells being bruised can start to produce ethylene. And the ethylene emanates from those bruised cells on the fruit. And that can trigger rapid fruit ripening of tomatoes, let's say, in the near vicinity, in the same box or in the same crate. And when they start to do it, they start producing ethylene as well. And so we get a chain reaction, and you can have rapid ripening of this crop. And so what they'll often do with tomatoes and other crops, I believe they do this with apple quite a bit as well. A, they refrigerate to slow down respiration in the first place. But B, they'll often pump CO2 or even nitrogen gas into the shipping containers. If there's nothing but CO2, or even if the plant does produce, or the fruits in this case do produce ethylene, respiration rates and therefore ripening will not be possible because there's a lack of oxygen. On the other hand, some fruits are actually intentionally triggered into ripening down the road. Perhaps maybe they take these tomatoes, reach their destination, and then they can either pump ethylene into the containers or use a spray like I was talking about with cotton harvesting earlier to produce ethylene and to actually accelerate the ripening process. So that wraps it up for what we call the endogenous hormones. There are a few other hormones that are prominent in certain species but not others, but these 5 hormones are what we consider to be the endogenous hormones. And just a quick review, I expect you to be able to tell me which are growth promoting and which are growth inhibiting. So here's how the list goes. First, we had auxins, and they are considered to be growth promoting and growth inhibiting. I'm just going to use P's and I's here. Then we talked about the gibberellic acids, which are generally thought of as being growth promoting. Cytokinin, based on the word for cell division. Promotes just that, and so we consider it promoter. Then we had abscisic acid, or ABA for short, which is considered inhibitory. And then finally we had ethylene, which is also considered inhibitory. And so plants use these 5 endogenous hormones many times in combinations with each other. There can often be responses that are a result of combinations of 2 or maybe even 3 of these compounds. And these chemicals are what most plants use to control virtually all of their growth responses to changes in the environment, to different times of the year in terms of light conditions or in terms of temperatures that they encounter, because plants lack a nervous system and muscles to be able to avoid those sorts of obstacles that they may face. And so they're basically stuck where they are for the most part, and they have to send signals to make preparations to go dormant, for example, or to maybe drop these leaves to conserve nutrients or to conserve moisture during periods of drought or extreme cold. And these are the 5 hormones that they use to do that.